Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Archibald Vivian Hill, a name synonymous with groundbreaking investigations into the physical processes underpinning biological function, embarked on a scientific journey that began with the fundamental question, how does muscle work? His early research, conducted in the late 19th and early 20th centuries, centered on the problem of understanding exactly how muscles convert chemical energy into mechanical work, a process that, at the time, remained largely a mystery. The challenge lay in quantifying the energy transformations occurring within muscle tissue, specifically the heat produced during contraction. This was a problem of immense complexity, demanding both innovative experimental techniques and a rigorous, quantitative approach far removed from the descriptive physiology that dominated the field. The prevailing understanding of muscle function at the time was largely qualitative. Scientists knew that muscles contracted and that this contraction was fueled by some form of energy, but the precise mechanisms and the energetic costs remained obscure. The challenge was not merely to observe muscle contraction, but to measure the energy released during the process with both accuracy and temporal resolution. Hill recognized that heat was a key indicator of the energy transformations at play. This was because, according to the first law of thermodynamics, energy is conserved, and the work performed by muscle, along with any other form of energy transformation such as electrical, must be accounted for in the total energy budget. Heat, therefore, offered a window into the energetic cost of muscle activity. However, measuring this heat presented formidable technical hurdles. The heat produced by contracting muscle is small, transient, and rapidly dissipated. Standard calorimetric methods, while adequate for measuring heat from large-scale chemical reactions, were simply not sensitive enough to capture the subtle thermal changes associated with muscular activity. Hill's solution, born of ingenuity and meticulous experimental design, involved the development of exquisitely sensitive techniques. He employed thermocouples, devices that convert temperature differences into electrical voltages to measure the minute changes in temperature associated with muscle contraction. These thermocouples, crafted with precision, were coupled with rapid calorimetry techniques. He developed a system that could measure the heat produced by a muscle in fractions of a second. This was crucial because the heat production during contraction is not a steady-state process. It fluctuates rapidly. The precise timing of heat release was central to understanding how energy flows through the muscle during contraction and relaxation. The experimental setup was designed to minimize heat loss and to ensure that the heat produced by the muscle was accurately captured and measured. This required careful insulation, precise temperature control, and sophisticated methods for calibrating the measuring instruments. The initial findings from these experiments were striking, and they challenged the prevailing understanding of muscle energetics. Early experiments, for instance, revealed that heat production occurred in two distinct phases: an initial burst of heat during the contraction itself, followed by a slower, sustained phase during relaxation. He demonstrated that a significant portion of the energy expenditure occurred during the initial contraction phase. This observation challenged the then current view, which had largely assumed that the primary energy consumption occurred during the recovery phase. This was a critical shift in perspective, moving the focus from the recovery phase to the contraction phase. Moreover, Hill's work demonstrated a clear relationship between the amount of work performed by the muscle and the heat produced. He showed that the efficiency of muscle, the ratio of work output to energy input, was far from perfect, highlighting the inherent inefficiencies in biological energy conversion. The implication of these findings was profound. It suggested that muscle contraction was a complex process, not a simple conversion of chemical energy into mechanical work. There were many factors at play, many steps, and each step had its own energetic cost. The significance of Hill's early work extended far beyond the specific findings. His approach represented a paradigm shift in the study of muscle physiology. He moved away from mere observation and description toward a rigorous, quantitative analysis. He insisted on precise measurements, careful controls, and the application of physical principles to understand biological phenomena. This approach, which would later become a hallmark of his research, set a new standard for the field. He was concerned with the precise measurement of heat production, the meticulous control of experimental conditions, and the careful interpretation of results. The influence of his teacher, John Langley, is clear in Hill's early scientific trajectory. Langley, a prominent Cambridge physiologist, instilled in Hill a deep appreciation for experimental rigor and a keen interest in the workings of the nervous system and the mechanisms of physiological control. Langley's own work on the autonomic nervous system, which involved precise experimental techniques and a focus on quantitative analysis, provided a model for Hill's later research. He learned from Langley the importance of meticulous experimentation, the value of careful observation, and the need to develop quantitative methods to probe the complexities of biological systems. This early mentorship was critical in shaping Hill's scientific approach and providing the foundational knowledge and skills that would enable his later groundbreaking discoveries. The emphasis on quantitative analysis and the application of physical principles directly learned from Langley would become fundamental to Hill's research program. This early influence was instrumental in shaping Hill's scientific trajectory and preparing him for the challenges of studying muscle energetics. The foundations of Hill's scientific investigations, established through his apprenticeship with Langley, provided a robust framework for his subsequent explorations into the mechanics of muscle. The challenge that gripped Hill was a complex interplay of energy, force, and velocity within contracting muscle fibers. Prior to Hill's work, understanding of muscle function was largely qualitative, lacking a comprehensive quantitative model to describe the intricate relationship between these parameters. Researchers grappled with the problem of how muscle, a biological machine, converted chemical energy into mechanical work, a process that demonstrated a remarkable efficiency, but remained elusive in its precise mechanics. Hill sought to transform this qualitative understanding into a precise, mathematical description. Hill's approach, echoing the physicalist leanings of the time, involved applying the principles of physics, particularly thermodynamics, to biological systems. His work was deeply influenced by the legacy of Hermann von Helmholtz, whose pioneering studies on the conservation of energy provided a crucial foundation. Helmholtz demonstrated that energy could neither be created nor destroyed, but could only change form. This principle provided the theoretical bedrock for Hill's investigations. Helmholtz's meticulous experiments, which quantified the energy transformations in various physical systems, provided a crucial precedent for Hill's ambition to measure and model the energy exchanges within muscle. Hill sought to quantify the energy used by muscle during contraction and relaxation, and to relate this to the force generated and the speed of shortening. The culmination of Hill's efforts was the development of the now celebrated Hill equation, a mathematical model that describes the relationship between force, velocity, and heat production in muscle. This equation, a cornerstone of muscle physiology, provided a predictive framework for understanding muscle behavior under a variety of conditions. Hill's equation, expressed as a hyperbolic function, elegantly captured the observed relationship between force and velocity during muscle contraction. The equation's formulation is straightforward, yet its implications are profound. It describes the velocity of shortening, represented by v, as a function of the force exerted by the muscle, denoted by p. The core of the equation is expressed as v equals b multiplied by p0 minus p divided by p plus a. Here, p0 represents the maximum force the muscle can generate, and b are constants that characterize the muscle's properties. The constant reflects the force at which the velocity is half the maximum velocity, and the b constant is the maximum shortening velocity. The equation's simplicity belies its power. It captures the essential features of muscle mechanics. To validate and refine his equation, Hill conducted a series of ingenious experiments. He employed a sophisticated apparatus that allowed him to meticulously control and measure the force, velocity, and heat production of isolated muscle preparations, typically for sartorius muscles. These experiments involved carefully monitoring the muscle's behavior under various loads and contraction speeds. He measured the heat produced by the muscle during contraction and relaxation, providing the data needed to calculate the energy expenditure. These measurements were then correlated with the force generated and the velocity of shortening. Hill's experimental design was meticulous, accounting for factors such as muscle temperature and the time course of contractions. His approach was a model of experimental rigor, demonstrating a profound understanding of the importance of precise measurement and careful control. The applications of Hill's equation were immediately apparent. It provided a means to compare the mechanical properties of different muscle types. Fast-twitch muscles, designed for rapid movements, were found to have different A and B constants compared to slow-twitch muscles, which are specialized for endurance. The equation also allowed researchers to predict the energy cost of muscle contraction under various conditions, providing insights into the efficiency of muscle work. For instance, the equation could be used to predict the energy expenditure during a particular movement, such as lifting a weight or running at a specific speed. The equation's predictive power extended to various physiological contexts, from understanding the mechanics of locomotion to analyzing the efficiency of muscle performance during exercise. Hill's equation continues to be a fundamental tool in muscle physiology and biomechanics. Its influence resonates in contemporary research, where it forms the basis for more complex models of muscle contraction. The equation, or variations of it, is still widely used to analyze muscle function, to understand the effects of fatigue, and to predict muscle performance in various activities. It has been adapted and extended to account for the complex behavior of muscles in different species and under various physiological conditions. The enduring relevance of Hill's equation is a testament to its elegance and its ability to capture the essential features of muscle mechanics. The impact of Hill's work extended beyond the immediate application of his equation. It provided a framework for understanding the energetic cost of muscle contraction. Hill's work emphasized the importance of considering both the mechanical and energetic aspects of muscle function. This holistic perspective shaped future research in the field. The equation spurred further investigations into the molecular mechanisms of muscle contraction, ultimately leading to a deeper understanding of the processes that drive muscle function. The Hill equation directly influenced subsequent models of muscle contraction. Huxley, a prominent figure in the field, built upon Hill's work to develop his sliding filament theory of muscle contraction. Huxley's model, which described the molecular interactions between actin and myosin filaments, provided a mechanistic explanation for the force-velocity relationship described by Hill's equation. Huxley's work, in turn, was influenced by the quantitative approach pioneered by Hill and his emphasis on precise measurement and mathematical modeling. The legacy of Hill's work is evident in the contributions of many researchers. In particular, the work of R. Niedergert, who further refined the understanding of the sliding filament mechanism, was informed by Hill's work on muscle energetics. Niedergert's studies on the structural changes within muscle fibers during contraction relied on the quantitative framework established by Hill. This demonstrates the pervasive influence of Hill's work on the development of a sophisticated understanding of muscle physiology. The equation contributed a durable framework for exploring the intricacies of muscle function. Archibald Vivian Hill's meticulous approach to understanding the complexities of muscle function extended beyond the mechanics of contraction. His work on oxygen debt, a phenomenon observed after intense physical exertion, represents a pivotal contribution to exercise physiology. The central problem Hill addressed was the metabolic puzzle of muscle recovery, how the body replenished its energy stores after strenuous activity, and what processes dictated the delayed oxygen consumption characteristic of this phase. Hill's experimental strategy, rooted in his earlier quantitative methods, involved careful measurements of oxygen consumption. He meticulously tracked oxygen uptake in humans and animals before, during, and after periods of exercise. This precise data gathering was central to his investigation. He utilized a variety of techniques to measure oxygen consumption, including the use of a spirometer, a device that allowed him to accurately quantify the volume of oxygen consumed by an individual. He also employed sophisticated gas analysis methods to determine the composition of inhaled and exhaled air, thus providing a comprehensive picture of oxygen exchange. This rigorous methodology enabled him to observe a consistent pattern. Oxygen consumption remained elevated for a period following the cessation of exercise, even after the initial demands of muscle contraction had subsided. This elevated oxygen consumption during recovery, the oxygen debt, became the focal point of Hill's investigations. He sought to correlate the observed oxygen consumption with the removal of metabolic byproducts accumulated during exercise. The prevailing understanding at the time offered a limited explanation. The scientific community was grappling with the intricacies of energy metabolism in muscle. Hill proposed a groundbreaking hypothesis, the oxygen debt was, in large part, linked to the removal of lactic acid, a byproduct of anaerobic glycolysis, a metabolic pathway that provides energy when oxygen supply is insufficient. This hypothesis was not immediately accepted. It challenged existing views. Hill's experimental data provided compelling support for his ideas. He observed that the rate of oxygen consumption during recovery correlated with the duration and intensity of the preceding exercise. The longer and more intense the exercise, the greater the oxygen debt. He also found that the rate of lactic acid removal from the blood mirrored the rate of oxygen consumption during recovery. This close correspondence provided strong evidence for the link between lactic acid metabolism and oxygen debt. He demonstrated, through careful experimentation, that the elevated oxygen consumption was used to convert lactic acid back into glucose, a process that required energy and, thus, oxygen. One specific example of Hill's work involved studying the oxygen consumption and lactic acid production in frog muscles. He subjected these muscles to various stimuli, inducing contraction. He then meticulously measured the oxygen uptake and the concentration of lactic acid in the muscle tissue. His data demonstrated a clear relationship that during the recovery phase, oxygen consumption increased and the lactic acid levels decreased. The recovery process, therefore, was a metabolic expenditure requiring oxygen to restore the muscle's energetic state. This work was published in a series of papers in the 1920s and 1930s, and these publications are still considered foundational in the field. The implications of Hill's research extended beyond the laboratory. His findings significantly advanced the understanding of exercise physiology. His work quantified the metabolic demands of exercise, providing a framework for understanding how the body responds to physical activity. The concept of oxygen debt explained the phenomenon of delayed muscle fatigue and the importance of recovery periods in athletic performance. His work laid the groundwork for many subsequent studies on muscle fatigue, training methodologies, and the physiological responses to various types of exercise. The modern relevance of Hill's work on oxygen debt is undeniable. The concept remains a cornerstone of exercise physiology. It is applied in sports science to design training programs, optimize athletic performance, and prevent overtraining. In medicine, the understanding of oxygen debt is crucial in the treatment of patients with cardiovascular or respiratory conditions. The principle of oxygen debt informs the design of rehabilitation programs and helps clinicians to understand the metabolic demands of various physical activities. The work continues to influence the study of metabolic processes. Hill's work on oxygen debt built upon the foundation laid by earlier researchers. Specifically, the work of Otto Mayhoff exerted a significant influence on Hill's understanding of the metabolic processes during muscle contraction and recovery. Mayhoff, a German biochemist, had made significant contributions to understanding glycolysis, the metabolic pathway by which glucose is broken down to produce energy. Mayhoff's work elucidated the role of lactic acid as an end product of anaerobic glycolysis in muscle. Hill built upon this knowledge. He integrated Mayhoff's findings in glycolysis with his own meticulous measurements of oxygen consumption to develop the concept of oxygen debt. This integration was a critical step in understanding the complex metabolic processes that occur during exercise. The influence of Hill's work on oxygen debt extended to subsequent generations of researchers. His findings provided a framework for further investigation into the intricacies of muscle fatigue and recovery. Many scientists expanded upon Hill's ideas, exploring the specific mechanisms involved in the removal of lactic acid and the resynthesis of energy stores. This work has helped to refine the understanding of muscle physiology. Hill's work provided a critical foundation for researchers who pursued a deeper understanding of the molecular mechanisms that underpin muscle function. Building upon his foundational work in muscle physiology, Archibald Vivian Hill made pioneering contributions to another crucial area of biological science, nerve physiology. His investigations into the speed of nerve impulses stand as a testament to his meticulous experimental approach and his profound understanding of the fundamental principles governing biological systems. The central scientific problem that occupied Hill's attention in this domain centered on quantifying the velocity at which signals traverse nerve fibers. This inquiry represented a significant challenge. The speed of electrical transmission in nerves is exceedingly high, demanding highly sensitive and precise measurement techniques. Hill's approach to tackling this problem was characterized by a blend of ingenuity and technical prowess. He drew inspiration from the work of Hermann von Helmholtz, who had previously demonstrated the feasibility of measuring nerve impulse velocity. Helmholtz's pioneering experiments, conducted in the mid-19th century, had established that nerve impulses, contrary to earlier assumptions, did have a measurable speed. Helmholtz's work served as a crucial starting point, providing the initial impetus for Hill's own investigations. Hill, however, sought to refine and expand upon Helmholtz's methods. He employed a range of sophisticated instruments, including sensitive galvanometers and, later, oscilloscopes. These devices allowed him to measure the minute electrical currents generated by nerve impulses and to determine the time it took for these signals to travel along nerve fibers. The galvanometer, a device for detecting and measuring small electric currents, served as a crucial tool in the early stages of Hill's experiments. It enabled him to detect the arrival of an electrical signal at a specific point along a nerve. However, the galvanometer's inherent limitations, particularly its relatively slow response time, presented a challenge in accurately measuring the extremely rapid transmission of nerve impulses. The introduction of the oscilloscope marked a significant advancement in Hill's experimental capabilities. The oscilloscope, an instrument that provides a visual display of electrical signals over time, offered a much faster response time than the galvanometer. This enhanced speed allowed Hill to precisely measure the time it took for an impulse to travel a known distance. The oscilloscope provided a visual representation of the electrical activity, enabling him to directly observe the time delays involved in nerve conduction. He could accurately measure the time elapsed between the stimulation of a nerve and the detection of the resulting electrical signal at a distant point. Hill's experimental setup typically involved the careful dissection and preparation of nerve fibers, often from frogs or other suitable animal models. He would stimulate the nerve at one point and then measure the electrical response at another, precisely known location along the fiber. By meticulously measuring the distance between the stimulation and recording sites, he could calculate the speed of the nerve impulse by dividing the distance by the measured time. The challenges inherent in these experiments were considerable. Maintaining the integrity of the nerve fibers, ensuring accurate timing measurements, and accounting for potential sources of error required a high degree of experimental skill and attention to detail. Hill's success in overcoming these challenges underscored his dedication to rigorous scientific methodology. He was a master of experimental design, meticulously controlling variables in a accounting for potential sources of error. Hill's measurements revealed that the speed of nerve conduction varied depending on the type of nerve fiber. He discovered that larger, myelinated nerve fibers, which are insulated with a fatty substance called myelin, conducted the impulses much faster than smaller, unmyelinated fibers. This observation helped to elucidate the relationship between nerve structure and function. The presence of myelin, acting as an electrical insulator, facilitated the rapid propagation of the electrical signal through a process known as saltatory conduction, where the impulse jumps between gaps in the myelin sheath. Specific examples of Hill's findings include his measurements of nerve impulse velocities in various types of nerves. He documented the differences in conduction speeds between different nerve fibers, providing quantitative data that substantiated the qualitative observations of other researchers. His work provided a firm foundation for understanding how the nervous system transmits information. The modern relevance of Hill's work on nerve impulse velocity remains profound. His measurements provided fundamental data that is still taught and used in neurophysiology. The concepts and techniques he developed continue to inform research on the nervous system. Understanding the speed of nerve conduction is crucial for comprehending how the brain and other parts of the nervous system process information and coordinate bodily functions. His work laid the groundwork for subsequent investigations into the mechanisms underlying nerve impulse propagation. Hill's work directly influenced subsequent research on the nervous system. His meticulous measurements and the conceptual framework he established paved the way for further inquiries into the biophysics of nerve conduction. His work was a catalyst for other scientists to explore the intricacies of nerve function. His precise measurements provided the groundwork for future discoveries. The impact of Hill's work is clearly evident in the research of Alan Hodgkin and Andrew Huxley, who built upon his foundational insights to develop the groundbreaking Hodgkin-Huxley model of the action potential. This model, a cornerstone of modern neurophysiology, describes the ionic currents that underlie the generation and propagation of nerve impulses. Hodgkin and Huxley's model, which earned them the Nobel Prize in Physiology or Medicine in 1963, provided a detailed mathematical description of the electrical activity of neurons, building directly on the quantitative data and conceptual framework established by Hill. The Hodgkin-Huxley model, which explains how electrical signals are generated and transmitted in nerve cells, relied heavily on Hill's earlier work on nerve impulse velocity. The foundation Hill provided was essential to the development of the model. Archibald Vivian Hill's legacy extends far beyond the specific equations and experimental setups that bear his name. His influence, rather, is woven into the very fabric of modern physiology and biophysics. He exemplifies the power of a rigorous, quantitative approach to unraveling the complexities of living systems. His work, a testament to the value of precise measurement and mathematical modeling, transformed the way we understand biological processes. The Nobel Prize in Physiology, or Medicine, awarded to him in 1922, serves as a formal acknowledgement of his groundbreaking contributions. Yet, the true measure of his impact lies in the enduring influence of his methods and ideas. Hill's focus on quantifying biological phenomena provided a framework for others to build upon. His work established the foundation for a more mechanistic understanding of life, shifting the focus from descriptive observation to precise, measurable parameters. His impact on the field of muscle physiology is undeniable. His work on heat production during muscle contraction, for instance, provided fundamental insights into the energetic processes driving movement. The Hill equation, a mathematical model describing the relationship between force and velocity in muscle, continues to be a cornerstone of muscle research. It is a testament to the enduring power of his quantitative approach. This equation, though developed nearly a century ago, is still used today in biomechanics and exercise physiology. It enables researchers to analyze muscle function and predict performance in various physiological conditions. The influence of Hill's methods is evident in the evolution of experimental design. His meticulous approach to experimentation, characterized by precise measurements and careful control of variables, became a standard for scientific inquiry. His use of sensitive instruments, such as the thermopile, allowed him to detect minute changes in heat production, providing critical data for his analyses. This commitment to precision set a new standard for physiological research, influencing generations of scientists. Hill's influence on the development of the field of biophysics is equally profound. His work bridged the gap between biology and physics, demonstrating how physical principles govern biological processes. His exploration of the energetics of muscle contraction and nerve impulse transmission laid the groundwork for the application of physical methods to the study of life. This interdisciplinary approach, now a defined characteristic of biophysics, can be directly traced to his pioneering work. The rise of quantitative biology, a modern field that emphasizes the use of mathematical and computational tools to analyze biological systems, owes a substantial debt to Hill. His emphasis on mathematical modeling and the development of equations to describe biological phenomena anticipated this trend. The Hill equation, for example, represents an early, successful attempt to model a complex biological process mathematically. His work established a precedent for the use of quantitative methods in the study of life. The impact of Hill's work extends beyond specific scientific discoveries. He cultivated a scientific ethos that valued rigor, precision, and the pursuit of fundamental understanding. This ethos has shaped the scientific community, encouraging a commitment to evidence-based inquiry and the rigorous testing of hypotheses. His influence is seen in the training of scientists. The focus on quantitative methods and experimental precision is a direct reflection of his legacy. The significance of Hill's work in the context of the rise of quantitative biology is paramount. His approach, emphasizing the use of mathematics and physical principles to understand biological phenomena, was a crucial step in this evolution. His work provided a framework for the application of mathematical modeling and the development of equations to describe complex biological processes. This approach, which continues to be influential in contemporary biology, is a hallmark of his legacy. Hill's influence extended beyond the laboratory. He was a strong advocate for the importance of scientific research and its role in society. He understood the need for scientists to communicate their findings effectively and to engage with the broader public. His commitment to public engagement helped to foster a greater appreciation for science and its impact on human life. His contributions to the understanding of nerve function, While perhaps less widely known than his muscle work, were nonetheless crucial. His precise measurements of nerve impulse velocity provided fundamental insights into the mechanisms of nerve conduction. This work, which laid the groundwork for future discoveries, demonstrates his versatility as a scientist. Hill's legacy is not merely a collection of scientific achievements. It is a testament to the power of rigorous inquiry, the importance of quantitative methods, and the enduring value of interdisciplinary approaches to scientific problems. His work continues to inspire scientists. His dedication to precision and the pursuit of fundamental understanding serve as a model for scientific excellence. His influence on the field of physiology and biophysics, as well as in the broader scientific community, is undeniable. His contributions have shaped the way we understand the fundamental processes of life. The impact of his work persists. His contributions have become integrated into the core of modern scientific thought. His work has influenced countless scientists and continues to shape the direction of research in physiology, biophysics, and related fields. His legacy is secure not only in the textbooks, but also in the ongoing pursuit of knowledge. Archibald Vivian Hill's influence remains a powerful force in the scientific world.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.